Hello everyone. Have you ever think about these kind of questions in your daily life? Why can some people drink milk but others get stomach pain? Why do chopped apples turn brown but a squeeze of lemon prevents it? Why do we need to refrigerate food to slow spoilage? All these kind of questions are related to enzyme. So today, I'm going to introduce the function of enzyme and an experiment about how to measure the enzymatic activity. So first, let's have a brief introduction. What is enzyme? Enzymes are biological catalysts that speed up reactions by lowering activation energy. So most of the enzymes are proteins. However, some RNA molecules can also be as enzyme, which are also called ribozyme. The enzymes speed up biochemical reactions without being consumed. Here, this is a comparison between the catalyzed rate by the enzyme and uncatalyzed rate of a reaction in our human body. Here we show that the catalyzed rate with enzyme are much more faster than the uncatalyzed rate, which means that the enzyme have a high efficiency. So how enzymes work? Here I would like to introduce two concepts. The first one is the substrate specificity. The substrate is the reactant that an enzyme acts on. So here, the red one is the substrate. Why we call it substrate specificity? It's because the enzyme can only combine with one kind of substrate in their active site and combine to produce the enzyme substrate complex. So the second concept is the induced fit. Induced fit of a substrate brings chemical groups of the active site into positions that enhance their ability to catalyze the reaction. Here we see the substrate's shape and the enzyme's active site's shape are really different. However, when they contact together, the enzyme's active site's shape will change and suit the substrate's shape and then combine together to produce the products. This is the substrate, they combine with the enzymes, and here comes the products. How to speed up enzyme reactions? I would like to divide them into two aspects. The first one is the substrate concentration. With an increasing substrate concentration, the speed of the enzyme reaction will also increasing. However, when all enzyme molecules have their active site engaged, the enzyme is saturated. If the enzyme is saturated, the reaction rate can only be sped up by adding more enzyme. Then the second aspect is about the local conditions. General environmental factors including the temperature and also pH. In most of the time, a higher temperature means a higher enzyme reaction rate. However, for the pH, it depends on what kind of enzyme and what reaction they are made. Also, chemicals that specifically influence the enzyme will also work. Now let's move on to the second part, lab experiment. Here I would like to introduce an experiment about how to measure the enzymatic activity. Here's the concept of the experiment. The first part is about how to draw a standard curve. Many enzyme assays produce a colored product whose absorbance at a chosen wavelength is proportional to its concentration. By preparing a set of non-product concentrations and measuring their absorbance, a standard curve, which is the absorbance versus concentration, will be built. This curve will fit a linear equation, and then it comes to the second part, which is calculate the enzyme activity. So for an enzyme reaction sample, you measure the absorbance. Use the fitted equation to solve for the product concentration. Knowing the product amount formed and the reaction time lets you calculate the enzyme activity. Here, the enzyme activity means the product formed per unit time. In the first part, what is the standard curve? Here, this is in sample. The standard curve is a calibration graph created by measuring the absorbance of a set of known product concentrations and plotting x-axis as the concentration of products. Since this is a standard curve, all the solutions here are standard solution, which are known values. A y-axis is the absorbance. So why the absorbance can be related to the concentration? Here, I'd like to introduce the BIOL110 law. The equation is just as listed. A in this equation refers to absorbance, and epsilon can show the light absorbing ability of the matter. The L here means the path length, and C here means the concentration. Here, the experiment is using the same temperature and the same plate. The epsilon and L are constant. Thus, the law states the absorbance is directly proportional to the concentration of a light-absorbing molecule. So here, let's come to the experiment of the first part. These are the materials that will be used, and this is the plate reader, which will measure the absorbance at wavelengths. This is the micropeptide and the tips. This is 96 well plate. In the first step, you will make a standard solutions. Then, we will put the standard solutions in the 96 well plates. Since here we assume that we already know the wavelengths of the color of the solution, we will just use the plate reader and then come to this form with a relationship between the concentration and absorbance. Using this form, we can graph the concentration versus absorbance line, which is also the standard curve. Part 2. Calculate the enzyme activity. How to calculate using the curve? Just as we said before, with the BIOL110 law, which states that the absorbance is directly proportional to the concentration of a light-absorbing molecule. Thus, we will use the equation here to show the relationship between absorbance and concentration and calculate. These are the materials we will use, and this is the water bath, which help keep constant reaction temperature. So first, we will put the enzyme and substrate together with the buffer and then put them into the water bath. Here, I will emphasize the same temperature and pH, which is also the same condition. That is because, just as we mentioned before, the activity of the enzyme will be impacted by some conditions, such as these kind of factors. So then, after the enzyme catalyzed reaction, we will add the stop solution, and then these unknown solutions will be created. After measure their absorbance, we will get another form with the absorbance and concentration. However, here we may have 3 samples in one group since we want to decrease the systematic error and the random error and then avoid some mistake. We will then use the absorbance data we have here to value on the x-axis, which is considered as the concentration of the products. When we know the product's amount and the reaction time, we can calculate the enzyme activity. So here, after we learn about the enzyme, we can understand these questions in our real life. So why some people drink milk but others got stomachache? That is because of the function of lactase, which is a kind of enzyme to digest lactose. Why do chopped apples turn brown but a squeeze of lemon prevents it? That is because of the impact of pH. With a squeeze of lemon, the pH will be decreased, and in a low pH condition, the enzyme cannot act as usual, and the reaction rate will also be slowed down. Why do we need to refrigerate food to slow spoilage? That is also because of the impact on enzymes that influence the enzyme reaction. However, this time is because of the temperature. After my presentation, I would like to have a big thanks to my professor in BIOL110 and my classmates, especially my group mates. Thank you very much for listening.